Okay, good. Okay, so being recorded now, so everybody watch their language, right? Okay. Welcome, everybody. I'm glad to have this opportunity to talk to you about a lot of the stuff that the BSF has been supporting and our researchers have been doing with regard to trying to find some sort of treatment for Barth syndrome. And some of this you may know a little bit about, some of this you may not, some of this you may just want to be updated on. So if you got any questions, type them in. If it's something really urgent, ask, tell Shelley to, stop Matt and ask him this question. So I have no problem with that. It works a lot better if somebody interacts with me because it's hard to tell what people are confused about and need some um, more explanation about. So let's get into it. Um, Another thing, you should also consult. We have a great newsletter and website, and they have a lot of this information on it. And you can go back and you can look at, the newsletter. Especially, I've outlined all the 10 grants that were given out in this last cycle, and then Kate McCurdy's got a really wonderful article that sort of has the perspective from where she's come from, which is from the very beginning about how things have progressed from practically nothing to something really substantial, like I show here on this slide here. There's 7 therapeutic ideas that are currently being pursued that we know about. Quite frankly, there's actually an 8th one called Bendavia, which is being that is a compound being developed by a company in the Boston area. And I know a little bit about that, but I won't talk about that because it's really somebody else's baby, and we don't know that much about it. Of course, we didn't contribute any funding to that 8th idea, but we know something about it, and actually it should be really interesting to see how that plays out. That will take some time, though. Right now, I can count at least 7 therapeutic ideas for Barth syndrome. And they're listed here. Some of these you already are probably familiar with to some extent, and some we've only started to really support in the last year or two with regard to the BSF being involved and people asking for money to put these ideas through. And I put this in a sort of a chronological order of of how we knew about it, how I knew about it, and how they're coming about. And I'm going to talk about repurposed drugs, bezafibrate being the one that is on my mind right now. Exercise therapy, which Todd Cade has been doing for the last several years. Nutritional therapy, we'll talk Richard Kelly and the idea, and actually nutritional therapy. I'll explain more about this. That's actually going to be part of anybody's real therapy or treatment in Barth syndrome, because part of the answer to treating Barth syndrome has to be where the guys with Barth syndrome start to build up muscle mass, increase their fatigue levels, or lower their fatigue levels, and get just physically stronger. So that can only occur through nutrition as well. So that's always going to be an important part of any combined therapy or treatment. Enzyme replacement therapy with Barry Byrne. We'll talk about gene therapy with Barry Byrne. Enzyme replacement therapy, that's with some guy out in the Seattle area that we've been working with. Lipid replacement therapy, a guy in the Oakland area that we've been working with, and raw scavenging therapy, which is very new. If you were at the conference in 2014, there were many talks about the ROS, or reactive oxygen species scavenging, that might be important in Barth syndrome. So I put these in a sort of chronological order, historical order, from my point of view for the most part. So let's talk about repurposed drugs, and the one that's on our mind is bezafibrate. And this idea. Bezafibrate, for those who don't know about it, it's what's called a anti-hyperlipidemic. It's given to people who have high cholesterol, but actually more specifically high triglycerides. It's been given for 25, 30 years. So it's a drug that's been around a long time. Unfortunately, it's not available in the U.S. It's available almost everywhere else, but it, for some reason, was not marketed in the U.S. But early on, under the observation here, Doctors Mindong Ren and Colin Foon found that when they added this compound, this bezafibrate, to Barth syndrome cells, it reversed that very unique biochemical signature. Well, it says, Shelley, would it be possible to copy this? Sure, no problem. I'll make them. I'll send them to Shelley, and then she can send them out wherever she wants. Okay. A unique biochemical signature of Barth syndrome is the cardiolipin monolysis cardiolipin ratio. You don't have to know much about that, other than the fact that it's very unique to Barth syndrome. And what Foon and Ren found was that when you added bezafibrate, it reversed that signature. So that's interesting. It's a very unique signature. And so what is this compound doing to reverse that signature? And so we got involved with the NIH through the SMART program. We obtained orphan drug status from the FDA for this drug. And we're coordinating with a company called Tribute to get the drug in a placebo and do a study. Now, in the UK, Colin Stewart has gone further than we can do because it's an approved drug in the UK, and he's already got funding to do a study with bezafibrate on Barth syndrome. And all I know is that they're in the midst of getting that trial put together, and we hope to, with the US, add to that effort or be a part of that effort. But because of the differences, because it's approved in the UK but not in the US, we have to find out a way to work that, to do a combined trial. And we're trying to do that. So right now, we expect to try to get the FDA to allow us to do a trial with bezafibrate in the US. And like I said, Colin Stewart and the UK people are already there. Bezafibrate's approved in France, yes. Yeah, I have no problem. In fact, the person who brought the idea of bezafibrate to our attention was a French researcher at the INSERM in Paris, a guy named Jean Bastien, and his wife Fatima. And they actually used this compound in another, it's Bastin, Jean Bastien. And they actually use this compound to treat people with a different type of mitochondrial disease called carnitine palmitoyltransferase 2 deficiency, and they saw some good effects. And so and that's actually where Foon and Ren got the idea and treated our Barth syndrome cells with it. And so we've been I consult with John all the time about where to go and how to do this. So the real problem, it's not so much a scientific or even clinical problem, it's FDA, being able to do a trial in the U.S. with a compound that has not been approved. So but I take advantage of whoever wants to help us in this effort. And the NIH has been very generous in the SMART program that allowed us to get this far. And we just want to take it to the next level and find out. A real question is this useful? And it's a very safe compound. It's been around 25 years. So I'm very hopeful that it's going to be good to use. And recently we did some work with Jaja Kachua we actually had Jaja and Mindong Ren and Colin Foon do some work with the knockdown mouse and bezafibrate specifically. We actually contracted with them to do that. And they got some very wonderful results that are not published yet, but we're going to use to ask the FDA for permission. They got some really remarkable results. I just want to leave it at that I really think bezafibrate's got a really good future, and we want to push it as fast as we can to get the permission that we need to from the FDA. So if there's not much more to say on the bezafibrate, if anybody's got any specific question, I'll go on to the next one. Now, exercise therapy, Todd Cade, Washington University. Well, I don't know if Aldo's been on the Todd's program, but he had none. Okay, you might want to take a look at it. Yeah. I don't know. Yeah, you got to move. I don't know if they allow people with a transplant or not. It. I think I've talked to Todd about that, and watch days were being recorded. Okay, that's true. Okay. One of the things about any therapy that we're going to decide to test is are transplanted boys, um, eligible for these trials? And in my opinion, unless there's some reason not to, we're going to try to do that. But in some ways, it makes it a little bit more difficult to analyze the data once it comes out. So we could discuss that, perhaps one-on-one together more concretely. Okay, Albert, good. Okay, exercise therapy. Todd has received several grants from us and has an R1 grant to look at this therapy for Barth syndrome. And what he had done earlier on was put the guys on what's called a cardiovascular rehab program, and to see if that would increase their strength, their muscle mass, and some of the other characteristics that we associate with Barth syndrome, improve them. What he found out was it improved their outlook, their view of things, but it didn't really increase their strength. It didn't really increase their decrease their fatigue levels. So about a year and a half ago, he started on weightlifting as opposed to what he was doing before was called aerobic conditioning. This is, but weightlifting is actually different. It's resistance exercise training, and that primarily builds up a different type of muscle in the body. And he's looking at that currently to see if that's a better way or a more effective way of treating Barth syndrome. So that strength training is now under study, and actually he got a grant for 2014, so it's not a further grant application pending. He actually has a grant to continue that work. So to me, I think exercise therapy is going to be part of any treatment for Barth syndrome because you have to build muscle, and we sort of know how to do that actually with weightlifters and with bodybuilders. We sort of know how muscle is put on in the body. So adapt those techniques to a certain way, and I think that's going to be one way where we're going to expect to see real progress in Barth syndrome treatment. Nutritional initiatives, probably the oldest initiative that we've looked at, because muscle mass increase is probably going to be important for seeing something that's really effective. And of course, Richard Kelly and now Hillary Vernon at Hopkins have looked at this, paid particular attention to this. It's actually, nutritional therapy by definition is not part of the FDA's portfolio. It turns out it's in a different area. It's under nutritional supplements, so it doesn't go through the same sort of approval process like a drug would, or actually something like an exercise therapy would. But as you know, nutritional therapy, Richard Kelly has identified that many of the guys have a low level of arginine and prealbumin in their blood, in what's called the blood plasma. And he has pretty good idea why that's the case. And the idea is very simply to compensate for that lowering by giving you more in your diet, giving the Barth guys more in their diet. And that's being done, and not in a clinical trial sort of sense, but on an ad hoc basis. And but that's something that's actually going to be part of, like I said earlier on, it's always going to be part of an effective Barth syndrome treatment. When you go to approve a drug, where you're trying to say, is this effective for treating Barth syndrome, you have to establish certain parameters that you want to see improved. And those parameters tend to be those things that every Barth guy has. And I think almost every one of the Barth guys has problems with fatigue. So fatigue and endurance is something that we'll be measuring and monitoring. And we hope that any effective therapy, like bezafibrate therapy or exercise therapy, will improve that in some sort of, prove the value that you have with that. So, so those are the ones that we know a lot about because they've been around for a couple of years, and we're really plugged into that. Either I'm in it, or Richard Kelly, or Hillary, and Todd Cade. We know them real well. They're at the conference. Hillary runs the clinic now, so we're pretty well plugged in with that. These other ones are things that the researcher that started this idea is really running with the ball. And one of them, Michael Chin at the University of Washington, has been, and you probably saw him at the last conference, he has enzyme replacement therapy as his idea. And I don't know if you know much about enzyme replacement therapy. When there are several diseases, usually called lysosomal storage diseases, where a certain enzyme is missing in the body. And simply what the treatment for some of these diseases is to take that, make that protein, that enzyme. An enzyme is a protein, by the way. Take that enzyme, make it in a test tube, and then give it to the person through their veins, basically, into their bloodstream. And so it's the same idea, but for Barth syndrome. So what he's done, he has taken the tavazin gene and grown it up in culture, basically in E. coli, which is what you usually make it up, and then has been trying to use it to see if it can reverse Barth syndrome. And he showed some very interesting data about the protein that he's made, this recombinant protein, and some very promising results so far. Right now, he's taken what he's done, and he's moving on to the mouse model of Barth syndrome, which we have to test these ideas. And that mouse model becomes more and more important as we get further and further into any sort of therapeutic idea, testing any therapeutic idea. Enzyme replacement therapy is already used to treat some diseases called Gaucher's disease, Pompe, Hurler's disease. So there's precedent for this in other diseases. So that's why this is really very exciting. And like I said, Michael Chin at University of Washington is really pushing this forward. He's a dynamo. Every year he's asking for grants, and we're very pleased with the enthusiasm that he shows and the work that is being done. Gene therapy is something that Barry Byrne, if you were at last conference's talk, Barry gave the keynote address. And of course, Barry is, well, he's a physician that has been around Barth syndrome from very early, and he knows it very well. And of course, he also happens to be the director of the Gene Therapy Institute at the University of Florida called the PAL Gene Therapy Center. And so the idea is, Florence, where are we up in terms of time scale of the enzyme replacement therapy? I don't know, because there's a lot of problems that could come up with enzyme replacement therapy. When you make an enzyme, let's just go back there. When you make an enzyme recombinantly, you have to test it to make sure that it's pure and doesn't contain certain what are called toxins. And but I think within 5 years, probably even less than that, he could be asking for volunteers to try his therapy. And I think because enzyme replacement therapy is known in other diseases, it'd be a lot easier for the FDA to prove a trial in that regard, because we've seen it in other therapies. Okay, so let me just get back to gene therapy. Barry Byrne, University of Florida, one of the leaders in gene therapy field. And I think gene therapy in the next 10 years is going to take off. It's been delayed historically because of some bad press that really wasn't the fault of the actual developers of it. But the idea is very simple. If you have a problem with a gene and you know about it, give the gene back to the person, and will that fix the problem? And so that's the essence of therapy. It's a little complicated because instead of using a needle, you have to put the piece of DNA into a gene, put a piece of DNA into a virus called adeno-associated virus, AAV. Then you inject that virus in the person, and then you hope it goes to where it's supposed to go to give the greatest effect. So Barry actually has a planning grant from us to work this, along with the other diseases that he's working on, into an effective sort of program where not only Barth syndrome but other diseases, you can just put in the defective gene and then set it up and inject the people and see if it works, how well it works. Now, gene therapy has limited success historically, but like I said, there was a lot of problems about 10 years ago. And I think we've gone past that problem. And I think very seriously, gene therapy is going to be frontline treatment for many diseases because it's easy to slot in a new gene for each disease. And we belong to that group. Barth syndrome has certain characteristics that make it a good candidate to be one of the first gene therapies in the U.S. that's approved. Right now, there's only one gene therapy approved, and it's in Europe. And we're still waiting for more U.S. efforts to get on board. But most of the effort's done in the U.S. Now, we talked about enzyme replacement therapy, and lipid replacement therapy is like the flip side of that. This is being done by a guy named Robert Ryan at the Children's Hospital and Research Center of Oakland, California, across the bay from San Francisco. As you know, Barth syndrome has a defective tafazin gene, and tafazin affects cardiolipin. One of the other ways of perhaps fixing the problems is to give the Barth guys cardiolipin that's already been modified properly, to what we call tetralinoleal cardiolipin. And so instead of fixing the upstream problem, Ryan has said, well, let's just fix the problem that the enzyme actually affects what's called the lipid or the cardiolipin. And actually, he has a really interesting system where he can deliver lipids to different parts of the body in a thing called what's called a nanodisc. That's involved here. It's ND. It's abbreviated, and it's actually shown here on the slide. It's actually like an Oreo cookie, a sandwich of phospho. I think it's the green, the spheres are phospholipid, and the blue is this thing called apolipoprotein. And the idea is he could seed those nanodiscs with cardiolipin and give that to, well, the idea is to give it to people, but the first step will be to give it to the mouse model of Barth syndrome and see if that really affects Barth syndrome, the symptoms of Barth syndrome in the mouse. And of course, if it does, or even if it gives us a little hint of that, then here is another completely different way of, looking for a therapy for Barth syndrome individuals that, it attacks the downstream product of the gene defect. So that's really interesting and actually very complementary. You will see all these ideas pretty much. You are going to use the mouse as a model to prove whether or not this idea is worthy of, going into humans. And that mouse model BSF made and distributed or allowed its distribution to anybody in the world. And one of them was Robert Ryan. So it works out really well that the BSF's been in a position to help researchers help themselves in that regard. And the last one here, this is the ROS scavenging therapy, and it's a little bit complicated because it involves a thing called ROS, which stands for reactive oxygen species. And if you ever remember things called antioxidants, people talk about antioxidants. You got to have antioxidants to prevent cancer and so forth and whatever. That's a little too simplistic. Aldo says, yeah, okay. That's a little too simplistic. In fact, you need reactive oxygen species in the body to actually signal different things to go on. So you can't just, as a rule, eliminate or think to eliminate reactive oxygen species and feel better. In fact, we know that that's not the case. We know that the usual antioxidants that people have tried, like vitamin E and vitamin C, those general antioxidants don't seem to work in Barth syndrome. Richard Kelly has told me this many times. He's always, because that's one of the therapies they try in other mitochondrial diseases. But there is an interesting idea that a certain type of reaction oxygen species made in Barth syndrome cells may be a culprit or may be causing some of the symptoms. And Min-Lung-Ren and Colin Foon had published on this idea in 2012. At that point, it was just an idea. And he's got some, Ren and Foon have some really interesting data about how that could really be causative. But it's not as simple as just giving somebody a vitamin E pill or an antioxidant pill or eat a lot of blueberries like people do. It's going to be something much more specific, much more chemical. The paper by Wang et al., which is actually from Bill Poo's lab at Boston Children's, it shows some idea of this reaction oxygen species being potentially, if you can reduce that, you might have some therapeutic effect. So right now we have at least 2 grants that we gave in 2014 cycle, 2 grants that are looking at this idea very specifically. And I really think that this is, this is a completely, every one of these 7 things is almost completely different from one another. They don't really overlap in in what they're trying to do. Yet, in the end, we want them all, or most of them, to positively affect Barth syndrome individuals. So it may be the case that we might need a couple of these. Like I told you before, I think nutritional therapy or some sort of nutritional background is going to be important for any one of for any really effective Barth syndrome treatment that comes down the pike. So I just have backup slides here. So I really, that's the 7 we know about. Like I told you about an 8th called a compound called Bendavia. These slides actually I put together to help get some outside grants. This slide just shows that, we have the dark blue. This is what we've been giving out as an organization over the years for grants, the money to, and those people who got the grants asked for grants from the federal government, and that number is put on there. As you can see, we get like $4 or $5 for every dollar that we put in. So we just want to continue that, and this just shows publications that go on. That's one way we look at how things have been going along. And here's where we need money. So yeah, part of this talk was to help put together a fund to take these 7 ideas plus 1, maybe 8, and bring them to the stage at which we could do clinical trials. And when you get to the clinical trial stage, it gets to be much more expensive. And I was in pharmaceutical industry for 19 years, and the pharmaceutical industry, it's very interesting. I was in the drug discovery part of the pharmaceutical industry, so the very initial parts of things. And in a large pharmaceutical company, 1/3 of the money is spent at that stage, at that early stage, trying to identify, something that has the potential to work. 2/3 of the money is spent in a pharmaceutical company at the point at which you're starting to go into man, and that's why we needed to ask for more money from different donors or whatever to get So when these ideas come to the point at which you need to try them out and ask for volunteers, and that's actually where Aldo comes in and his brothers. They got to be available to test these ideas and volunteer for it. That's where it just costs a lot of money. We could spend a lot of time telling you why. Yeah, sure, more needles, right? Well, let's get away from the needle thing. Actually, some of these things may not be needle driven, but like the nutritional initiative. But for some reason, it just costs a lot more money. And it turns out there's fewer people in a pharmaceutical company that work at that expensive end. It's just that the money goes out to pay for clinical trials, which tend to be expensive. So that's you can read about these ideas in the newsletter. We have them on the website. If you look into the grants that are given to various people, you will see that. Matt, do you want to mention the LZZ696 trial? All right. LZC 696 is a dual-purpose compound that Novartis is putting out there, not for Barth syndrome. It's actually for heart failure and heart-related diseases, but it may be very effective in Barth syndrome. Now, John Jeffries is part of, I don't know if he's part of the approval process or he's involved with that. John Jeffries spoke at the meeting in last June at our conference. I don't really know what to say more about that, other than the fact that it should be, as it gets developed, as it gets approval, I think any cardiologist treating Barth guys will look at this and think, if one of those guys gets into a problem, this is a potential treatment that they should ask for. Now, even if it's not approved at that point in time, the individual cardiologist or physician that's treating a Barth guy, especially if they're in trouble metabolically, I think that's something that you will, it would be important for them to really perhaps consider. Lincoln clinical trial note, not a specific trial. No, it's not a specific trial. They're not going to tell you the truth, you probably wouldn't be on there for a clinical trial because, let me just tell you, this is from Novartis, and Novartis is my old company, it turns out. These indications are for, people going through heart failure. There are going to be trials of 3,000, 4,000 people. In fact, they're already on those trials. What's going to happen is this will be approved for the general condition of heart failure in probably elderly people. And what will happen is somebody like John Jeffries, who's treating a Barth guy, if that Barth guy is getting into problems, in a cardiac sense, he'll probably prescribe it off-label for him at that point. That's the way things really work, because nobody does a trial for Barth syndrome like we're trying to do. Nobody does a clinical trial to treat Barth syndrome individuals. You do it for a large indication, and then the physicians have the ability to prescribe drugs with a reasonable idea behind it to treat their patient, because that's ethically that's what they're trying to do. They're trying to take care of their patient. And but from the point of view of us, Barth guys volunteering for the trial, it probably wouldn't be in their parameters to test. That's why it's important to monitor the situation, see its approval process, make sure your cardiologist is aware of it, and that if the situation arises, he has that to consider in treatment, in acute treatment of what's going on. Hope that answers the question. I don't know. Is everybody back? Are they all muted? Looks like everybody's muted.
No, I do have them muted, Matt. And if anybody has a question, just let me know and I will unmute you. But I think one of the things that's really exciting for me is that we started out hoping that we would get to the point where we would have therapies, and here we are today, and it's it's in the horizon. We are following up with, as you say, partnered trials or drugs that have already been developed, and how might that benefit those who have Barth syndrome, and more targeted treatments. And one of the questions earlier was, where are we with this? And there was one point where we didn't know, and here we are within years, very few years, a handful of years, hopefully, for some targeted treatments.
Well, we're already pursuing treatments. I mean, the bezafibrate is going ahead in the UK, and they seem like it's going slow, but that's the way trials go. Todd Cade is doing exercise therapy at the moment, at this very moment. Uh, those are things that are on board. Of course, Richard Kelly has been looking at the nutrition of Barth guys, especially when they get into trouble, and been advocating, increasing those levels. These other ones, the gene therapy, lipid replacement therapy, enzyme replacement therapy, ROS scatter, they're going to be a little bit longer, but we have things now that we're testing that we need guys to volunteer for, that we need people to be part of the BRR, so they could be, directly, they can have their records directly looked at and say, all right, there's somebody we would like to see for a clinical trial. It turns out when you advertise for a clinical trial, you can't go pick people. They have to come to the clinical trial people of their own volition. What the BSF does, as you know, is try to make everybody know what's going on everywhere, so that the volunteers can volunteer when the time comes up.
And we have to, when they come to us, they want to know who is verified. And when somebody joins Barth Syndrome Foundation as a family member, we can't make their membership conditional upon having a copy of their genetic results. So with the registry, by having copies of those genetic results, we can say these people have been verified to have Barth, which is really important for investigations. And to go back to the Todd study, I did want to say that I spoke with him last week, and his greatest need is with the older guys. He believes that he has one other individual who's under the age of 18 who has signed up, which is the last person he needs. So the biggest challenge we have right now is with the young men who have Barth syndrome, and stressing the importance of participating, because we are such a rare disease that it's very difficult for us to receive NIH funding, because there's a sense that we wouldn't have enough participation. And if that happens, with us, it will really affect future funding. So it's really important that when we do have these studies where we need people to participate, that we get the numbers that we need.
Let me just add, Todd has an R01 grant to look at exercise therapy and Barth syndrome, but he only got it on the second try. On the first try, the reviewers said, we don't think you're going to get enough people. And then you and I tried to beat the bushes and got more people to volunteer. And then when he reapplied, he got the money for it. So yes, that's essential to any sort of clinical trial. The NIH is not going to fund things or help fund things. What? Who's typing like that?
Oh, I'm sorry. That's my dog drinking water.
It sounds like a horse. It sounds like a horse just went by your place. It's essential to have enough people, because the NIH is not going to fund something that has no chance of going ahead. So we got to always approach, when one of our researchers asks for a grant, we got to step up to the plate and say, when Todd says he's going to get 20 individuals, he could show them 20 different emails saying, I'm ready to sign up for the trial. And the same thing's going to happen for other trials too. And let me just add to this: Todd's studies are the backbone of all the future clinical trials, because he's looking at the parameters very, very carefully that we're going to use to show whether gene therapy is effective, whether enzyme replacement therapy is effective, lipid replacement, even bezafibrate. So helping Todd out helps everybody out. And it's the same thing. As soon as we get into the other clinical trials, what we'll discover is certain things that occur that we need to be aware of, but also things that perhaps we should alter clinical trials because of what we found in a previous clinical trial. So, people like Todd who have been doing the original work of clinical trial work are extremely valuable. And the data that he's developed, the data that he's published on already, the data he's going to publish, the stuff that Hillary Vernon collects at the clinic in Baltimore, those publications or soon-to-be publications are all important to help to design a clinical trial, make sure that it works out. Yeah, Dr. Cade has a very limited response to the over 18 crowd. Some of that I think may have to deal with a lot of guys over 18 have got a new heart. And I think his parameters were you can't be transplanted, but I think he may be able to alter that. That's not shocking. No, it's not. Because you want to set up an experiment so there's very little, you want to set it up so that when you interpret it, things are going to be very clear to your interpretation. And if you have a new heart and you have Barth syndrome, you certainly still have the problems with fatigue, but the problems with your heart are a little bit different now. You have different problems, but they're not the same sort of problems that you had before because it's a different heart. It's a genetically different heart. It doesn't have stuff. Right. Okay. Yeah. Shall we just put some stuff on? So, like I said, I wish every one of the Barth guys volunteers for every one of the trials because we don't have a lot of them, and we need to see as many as possible in these trials. And hopefully all these trials won't occur at the same time. That will, they'll be spread out. But they all help each other in trying to prove to the FDA what's a safe and effective treatment. And that's where we're at. And like Shelley said, we started out with very little for therapy that's specific for Barth syndrome, and now we have some very specific ideas that have a really good chance of making a big impact on the Barth guys. So, Shelley just said, any questions from anyone or concerns or anything at all? It's hot and humid here in New Jersey, but I don't think anybody's really concerned about that. I hope Paris was nice. One of
the things that we
unmute him.
One of the things that we often hear, or I often hear from families, is their concern about safety and participation, and there's a concern about when we get to the point of clinical trials, there is concern kind of in the back of folks' mind. I don't know if anyone wants to mention that or say it, speak to that themselves that's on this call, but I have heard that concern privately.
Well, every clinical trial is got to be approved by what's called an institutional review board, and that board is charged with not guaranteeing the safety or efficacy, but looking at the question of safety of any treatment that's being proposed. And it's a balancing act. In a drug therapy trial, drugs are not innocuous by definition. They do have effects, and some of those effects may not be good. Obviously, you want one of the effects to be something good for, let's say, the disease you're studying. But there could be other side effects that might make it, the cure worse than the disease. So in the early stages of a clinical trial, safety is paramount. In fact, most studies don't go to a clinical trial unless there's certain safety criteria that are used. Now, let's take, for instance, bezafibrate. It's been used in humans for 25 years. We're very knowledgeable about the safety of this product, so there's not much concern. A new entity like enzyme replacement therapy, though I think that's probably pretty safe too. It's newer. It's not been tried that much. We have a few diseases that's been tried on, but because of that experience, people know they think they know what to look for in the next clinical trial with the next enzyme replacement therapy. So we have a little bit more to go on, but it's still, kind of slow going because you want to know, want to look at those signs. If there's a problem, we want to know about it early so we don't endanger the the volunteer any more than we have to. Then we have compounds we never even heard of before, don't have any experience in the pharmacopoeia. So those are going to be ones where you're going to be very, very, uh, let's say, conservative because you don't know much about it. You know that when you give it to a rat, it doesn't die. Other than that, it's, you still, when you go into humans, it's, you're a little cautious and much more cautious. So it depends a lot on what the treatment is. Like Todd's exercise therapy, that has to go through an IRB as well. Well, we know exercise therapy is good. In fact, one of the slides I showed you that there's actually evidence that exercise therapy is good for a variety of diseases. So that makes us feel very confident that, when we do this therapy, it shouldn't be a problem with our Barth guys, at least not specific for that. So, that's how safety is addressed there. And then safety is addressed obviously by the people doing the study, but also this IRB, and usually also what's called a data safety monitoring board in any clinical trial. And that's like a star chamber where they look over the shoulder of everything. And if they see a problem, they identify it real quickly and either stop the trial or modify the trial. Sometimes they actually stop the trial because it's so good, don't need to do any more. That's happened like a couple times in my experience. But